Hello my wonderful biochemistry students. This is the first video presentation of the 2 video presentations I promised that I would give you guys while I was away here in Texas for a research conference. This chapter, of course, is chapter 19 from our biochemistry text, talking about harvesting electrons from the cycle. In my typical tradition, I want to begin by sharing you a few scumbag teachers of the day from quickmeme.com. I love this first one. Expects you to know the most complicated of theorems. Can't figure out how to turn on the projector. And the next one, this is cursive. You're going to use it for the rest of your life. Dang. Before beginning, I wanted, of course, share something interesting about chemistry history. These are molecules of the day: paclitaxel and 10-deacetyl-baccatin. Paclitaxel, known commonly as Taxol, is a mitotic inhibitor used in cancer chemotherapy, isolated from the bark of the Pacific yew tree. Because it takes about 1.3 tons of yew bark to get 10 grams of Taxol, and doing so kills the tree, an alternative means of obtaining large amounts of Taxol had to be developed. In time, a compound called 10-deacetyl-baccatin, shown here at the bottom, was found. It's made by the European yew tree. 10-deacetyl-baccatin can be harvested from the tree's needles, which grow back without killing the tree. In the late 1980s and early 1990s, Robert A. Holton, an organic chemist from Florida State University, devised and patented a way of synthesizing Taxol from 10-deacetyl-baccatin, thereby providing a scalable means of making industrial quantities with the anti-cancer medicine. You'll probably notice as you look between the structures of these two that they are structurally very, very similar, so you can imagine how the synthesis might have gone. Now, parenthetically, I've been told that a good chunk of Florida State's football team budget is paid for by the royalties from this patent by Robert A. Holton. I'm not sure if that's actually true, but I have heard it at least. Now, after this presentation, you guys should be able to know what the citric acid cycle is and does. Memorize all the names of the intermediates in the citric acid cycle and the basic reactions that occur between each intermediate. Now, I want you to note that I won't have you memorize the names of all the enzymes. Know how many ATP, NADH, and FADH2 molecules are produced by the citric acid cycle. Know what 3 enzymes are regulated to control the citric acid cycle and how they are regulated. Know how oxaloacetate is reformed once it's consumed. And know what the glyoxylate pathway is and what its purpose is. In our last lecture, which we had in class face-to-face last week, of course, we reiterated the fact that glycolysis converts glucose into pyruvate. We then learned that pyruvate can be used to make acetyl-CoA. We also mentioned that the resulting acetyl-CoA can be taken on through the citric acid cycle to eventually make more ATP. The citric acid cycle occurs in the mitochondria. Our last lecture focused on the conversion of pyruvate into acetyl-CoA, but we didn't talk in any detail about the actual citric acid cycle itself. We'll now fix that oversight. In this lecture, we'll talk about how acetyl-CoA gets incorporated into the citric acid cycle. Then we'll go step by step through the entire cycle. In the citric acid cycle, which is also called the Krebs cycle or the TCA, tricarboxylic acid cycle, acetyl-CoA's acetyl group, which has 2 carbons, is combined with oxaloacetate, which has 4 carbons, to make a 6-carbon molecule. This 6-carbon molecule then goes through a series of changes that ultimately produce 3 NADHs, 1 ATP, and 1 FADH2. Oxaloacetate is regenerated in the end to catalyze the next cycle. The NADHs and FADH2 can later be taken into something called the electron transport chain, which we'll discuss in a later chapter, to yield 9 more ATPs, which comes to 10 total if you include the ATP from this cycle. You should note that the cycle makes CO2 over here, which represents a net oxidation or burning of some of the carbons that came originally from glucose. So here's the whole crazy thing in one picture. Once again, you'll see that our acetyl-CoA, which was originally made from pyruvate, which was made from glycolysis, converting glucose, is combined with oxaloacetate to make citrate, and then citrate undergoes subsequent transformations all the way around the cycle to yield 2 or 3 NADHs, 1 ATP, and 1 FADH2. Once again, it regenerates the oxaloacetate to take another molecule of acetyl-CoA through the cycle. Now we'll go through the entire thing step by step in detail. In step 1, oxaloacetate, a 4-carbon molecule, and the acetyl group, which has 2 carbons in it, from acetyl-CoA are combined to make citrate, a 6-carbon molecule. This reaction is catalyzed by an enzyme called citrate synthase. So you'll notice that acetyl-CoA right here and citryl-CoA right here are both thioesters. They both have a carbonyl carbon bonded to a sulfur right here. So why in the world doesn't citrate synthase hydrolyze acetyl-CoA? So you'll notice that it only hydrolyzes citryl-CoA right here, but doesn't touch acetyl-CoA. So I hope you're at least wondering or thinking about that question. Here's the answer. Citrate synthase is shaped in a way that allows it only to bind with acetyl-CoA after oxaloacetate has been bound. Let's go back and take a look at that. So you can imagine the oxaloacetate having to be bound by citrate synthase first before citrate synthase is shaped in such a way that it allows acetyl-CoA to come in and combine with the oxaloacetate. If that weren't the case, then the citrate synthase would just hydrolyze acetyl-CoA instead of the citryl-CoA. This thereby allows citrate synthase to only hydrolyze citryl-CoA and not acetyl-CoA. In step 2, citrate is isomerized to form isocitrate, a reaction that's catalyzed by this enzyme called aconitase. You'll notice that this goes through this intermediate called cis-aconitase and essentially amounts to a dehydration and then a rehydration. In other words, an exchange of the placement of this hydrogen and this OH, going from starting material to product. In step 3, isocitrate, which once again possesses 6 carbons, is converted to alpha-ketoglutarate, shown here. In this process, isocitrate's C2 carbon, this one right here, gets oxidized up to a carbonyl carbon state, and NAD plus gets reduced to NADH. This forms this intermediate called oxalosuccinate, which is then decarboxylated. That is, we remove CO2 right here from the middle to make alpha-ketoglutarate. Decarboxylation is very exergonic, which helps drive this reaction forward. This reaction is catalyzed by an enzyme called isocitrate dehydrogenase. In step 4, alpha-ketoglutarate, which we just made, which has 5 carbons, is converted to succinyl-CoA, which has 4 carbons. In the process, alpha-ketogglutarate's C2 carbon, the one right here, gets decarboxylated. In other words, we release the CO2 that's colored here in blue, and then this carbon gets oxidized up to be a thioester again. NAD plus in turn gets reduced to form NADH. This reaction is catalyzed by alpha-ketoglutarate dehydrogenase complex. Now I realize that this may sound a little bit weird, but this is actually the place where I want to stop the lecture for today because we are halfway through the chapter. In my next video presentation, we will finish our lecture, the last 12 slides, to complete our discussion on the citric acid cycle.